Hello, everyone, and welcome to today's session. My name is Marta Dejia. I'm a SynGAP parent and director of the SynGAP Research Fund. We are very excited to continue the SRF SynGAP Research Fund webinar series. The goal of the series is to empower your communications with clinicians as you get more clear knowledge of SynGAP. We also want to give you a plug to our next presentation, improving the assessment of SynGAP1 and related genetic conditions by creating online measures for parents and patients, which will take place on Saturday, July 17 at 9 a.m. Pacific, noon Eastern, by Dr. Thomas Frazier. Our talk today is SynGAP modulates the body biological clock. What SynGAP1 mice can tell us about light and speed. I have the pleasure to introduce today's speaker, Dr. Sydney Aten. She obtained her PhD in neuroscience from the Ohio State University. She worked as a doctoral student in the lab of Dr. Zhou, researching basic astrocyte physiology and the effects of chronic stress on astrocytes gap junction coupling. Related to SynGAP, she is the first author on an important paper. SynGAP is expressed in the murine suprachiasmatic nucleus and regulates circadian-gated locomotor activity and light entrainment capacity. Her interest is researching the molecular cellular underpinnings of circadian clock, entrainment, cognition, and mood. She is also an aspiring as astronaut. A recorded version of this webinar will be available in the SRF website under webinars on the family menu. By the end of the presentation, you will have opportunity to get the answers to your questions. We love to hear from you. Please write your questions in the Q&A. For those of you just joined us, welcome. And again, our speaker today is Dr. Sydney Aten, and her talk is SynGAP modulates the body's biological clock. What SynGAP1 might can tell us about light and speed and sleep. Okay, and now it's my pleasure to turn things over to Dr. Aten. Hello, good morning.
Hi, good morning. Thank you so much, Marta and Mike, for allowing me to speak here today. I'm so excited. I think it's actually absolutely incredible what you guys are doing. So I'm so excited to give a talk on SynGAP. So let me share my screen. Oops. Let's see. Can you guys see that okay?
Yes.
Can you guys see my arrow or my pointer? Okay, awesome. Just wanted to make sure. All right. So I just wanted to give a brief outline on kind of my talk today. And when putting this presentation together, I kind of thought it might actually make a little bit more sense to work backwards a bit, going from the observation that kids with SynGAP, display these sleep abnormalities and have these sleep issues, and then kind of translate that to a potential mechanism as to why this could be, the case, why this could be happening. And so I'm going to give an overview of circadian clock timing, and then also note a really critical signaling pathway called the MAPK signaling cassette, which is really crucial in kind of regulating or modulating the circadian clock within your body. And then finally, like Marta mentioned, I'm going to give a brief overview of a recently published paper from our lab, just showing or detailing the expression and function of SynGAP within the body's master clock. So within that master clock that controls these circadian rhythms, again, throughout your brain and the rest of your body. So much of the literature in both humans and in mice has shown that SynGAP haploinsufficiency leads to sleep disruption. And I believe Dr. Kadam, I think she gave a webinar a while ago, just detailing how these SynGAP mice have increased incidence of seizures during these transition states, sleep dysfunction, hyperactivity, amongst other things. And I don't want to go into too much detail with respect to some of this more clinical data, because I believe that Dr. Holder also mentioned some of his published and unpublished data, again, showing that these kids with SynGAP have sleep abnormalities in addition to, disruption in sleep architecture, delayed onset of sleep, quality of REM sleep, etc. Furthermore, I'm not a sleep scientist. I've studied circadian rhythms, so I can't give too much input into that. But what I was able to do for one of the chapters of my dissertation was actually examine or provide these questionnaires to a SynGAP patient registry, noted as MRD5. And so here I just wanted to kind of highlight the prevalence of these, of the SynGAP kids who reported sleep issues. And so you can see here, you know, between 70 and 90% of these kids reported some sort of sleep issue, kind of again paralleling what we're seeing in the literature and also in these mouse models as well. Here I'm showing that, so if you look on the left, these are kind of de-identified patients or SynGAP kids from the registry that I was able to obtain. And then many of these children actually showed formal or had formal sleep diagnoses. One of them was irregular sleep-wake rhythm disorder, and the other being delayed sleep phase syndrome. So again, I thought that was interesting to kind of highlight that, you know, some of these kids are actually formally being diagnosed with some sort of sleep issue. And then finally, and again, I think this has probably been discussed or potentially been discussed in other webinars, but just the prevalence of sleep aids that are used by these kids with SynGAP. So I believe 20 out of 35 of the respondents reported using melatonin as a sleep aid. So I think that's about 60% of these kids are using melatonin in order to kind of combat these sleep issues that they're experiencing. And then finally, from this questionnaire, from these questionnaires that I was able to distribute, I was able to ask the questions, you know, are these kids being prescribed chronotherapy or medication for sleep issues such as melatonin? Are you having fragmented sleep, early wakening, trouble remaining asleep, trouble falling asleep, etc. And then I compared this data. So these SynGAP kids are in the green bars here, and you can see the percentage of individuals affected here at the bottom. But I did a really broad literature search and compared the number of SynGAP kids that are experiencing these sleep issues with individuals or kids who were diagnosed with autism spectrum disorder, and then also with typically developing individuals. And again, here I just wanted to highlight, you know, the prevalence, just how many of these SynGAP kids are experiencing these sleep issues, which again has been reported before. And so that kind of brings us to the question, you know, what might be contributing to some of these sleep disturbances? You know, and certainly epilepsy and these uncontrolled seizures that some of these kids with SynGAP have will play a role in altered sleep, especially if that's causing them to wake up at different times of the night. But I kind of wanted to get a little bit deeper into what else could be going on, what other processes could be contributing to some of these sleep abnormalities. And so I wanted to touch a little bit on the more conceptual framework for sleep-wake regulation within the body. And so one of the most common hypotheses of sleep-wake regulation is the idea that sleep is regulated by two main processes. The first being your circadian rhythm, and then the other process being this homeostatic drive or need for sleep. And so this first process, the circadian process, is what I'll talk about later. That's kind of the topic of the lecture. But essentially, it's built upon the idea that your biological clock is controlling circadian rhythms throughout your body. And that biological clock functions in response to light that you get from your environment. And thus, it's relatively independent of the amount of sleep that you've had and the amount of sleep that you need. Again, this process is more coordinated on that day-night light cycle. But these circadian rhythms alone, they're not sufficient to cause or lead to the need for sleep. And so there's also this propensity for sleep known as the homeostatic sleep drive that also occurs within organisms. And it's more of this biochemical system that kind of reminds your body, hey, I need to sleep. And it works rather intuitively in that the longer a person is awake, the greater that drive or that need for sleep becomes. And thus, the more likely that person is to actually fall asleep. And so if you look here on this schematic, you can see here this homeostatic sleep drive is indicated here by this solid line. And the circadian arousal drive is indicated by this more dashed line. And so again, you can see that as the day goes on, this individual is experiencing a greater need, or this homeostatic sleep drive is increasing. And that's kind of not combated, but it's kind of going against the circadian arousal drive. And you'll notice the circadian arousal drive increases throughout the day until the evening time point. And this time point here is where you see melatonin being produced. And when melatonin is produced, that actually allows the body to fall asleep. And so that can actually lead to this sleep onset. And then again, if we're following this homeostatic sleep drive, you'll see that that's decreasing throughout the night, indicated by this kind of like gray or dashed bars here. And that homeostatic sleep drive is decreasing throughout the night because, again, you're finally experiencing that sleep that your body was telling you that you needed to get. Until finally this point of waking occurs, because your circadian arousal drive kind of picks up again, and modulates these body systems that are, again, telling your mind, hey, I need to wake up. It's time to wake up. It's time to start your day, etc. So these are kind of the two main processes that are thought to underlie this sleep-wake regulation. And so knowing that these two processes can regulate sleep, this begs the question, how might SynGAP play a role in these processes? Or rather, why does SynGAP haploinsufficiency cause these sleep issues? And so we know that SynGAP regulates synaptic plasticity, and this process is really heavily involved in both epileptogenesis and in sleep homeostasis. And so here's a schematic that you've probably seen before. It was done or created after a seminal study published by Dr. Nier's lab. And essentially what it's showing is just that SynGAP is a really key modulator of AMPA receptor insertion at the synapse. And so you can see here on the left, so right here, this is just at baseline. So this is a cell or a synapse that has not been stimulated. And you can see here that SynGAP is kind of situated here in this postsynaptic density. And it's going to negatively regulate these signaling processes that are really tightly able to control AMPA receptor insertion at the surface. But then you'll see after LTP, which is long-term potentiation, it's a type of synaptic plasticity that can lead to learning and memory formation. You'll see that SynGAP is actually dispersed from the spine. So upon its phosphorylation or its activation, it actually leaves the dendritic spine. And that allows for this, the signaling pathway to produce or to lead to even more and more AMPA receptors being inserted here at the surface. And so again, I just wanted to kind of go through this schematic because it's showing that SynGAP is indeed a modulator of these AMPA receptors. And why is that important? Well, that's important because the synaptic homeostasis hypothesis of sleep-wake regulation, it posits that wakefulness, so when a person is awake, that tends to promote this idea of synaptic potentiation, so more neuronal activity, more synaptic activity, while during sleep, it favors synaptic depression or less activity. And so you'll see here in the schematic that AMPA receptors tend to be increased during this period of wake, and during this period of sleep, they tend to be decreased. And so again, this is really important because in SynGAP heterozygous animals, these SynGAP het animals likely have trouble kind of down-regulating these AMPA receptors potentially during this sleep period. And so if you have abnormalities in the ability to regulate this AMPA receptor and these AMPA receptors being inserted at the surface, that could also lead to sleep issues. And so that kind of covers the more, sleep homeostasis or homeostatic drive for sleep and how SynGAP may or may not be involved. But kind of where my story comes in, or where our lab story comes in, is might there also be a circadian component that involves SynGAP? And so I know I briefly, mentioned this a bit earlier in one of the first few slides, but I wanted to give some background on circadian clock timing. And so we know that Earth's 24-hour rotation allows for this diurnal cycle of light and darkness. And so for years and years, studies have shown that light is the most important or prominent, it's called Zeitgeber, or timing cue or stimulus that can set the phase of your circadian rhythms throughout your body. And so it's the circadian rhythm that coordinates these physiological processes throughout this 24-hour day. And in mammals, this major pacemaker for circadian rhythms is found within the SCN of the hypothalamus, or it is the SCN of the hypothalamus. And the SCN stands for suprachiasmatic nucleus. And again, this is serving as the body's master clock or master pacemaker. And so if you look at the schematic here on the bottom right, you can see this structure situated here kind of at the bottom of the brain, at the ventral side here, located in that hypothalamic region. And then here's another picture. So this is just a fluorescent image that I took. So this is a coronal mouse brain section, an entire brain section. And then here I boxed in the SCN. And so you can see that this SCN structure is kind of this bilateral nuclei. It's kind of teardrop in shaped. And so each one of these dots is just indicating, each one of these blue dots is just indicating a nucleus or a cell. but I just wanted to kind of show exactly what it's looking like, at least in a mouse brain. And so each one of these nuclei comprises approximately 10,000 neurons, which is really, really cool. And so how does this circadian, clock timing process work? Well, at the molecular level, this circadian timing system, it arises from this really well-characterized set of, interlocking, it's called a transcriptional translational feedback loop. And so it's this autoregulatory loop that allows for the generation of new circadian clock genes. And that then can in turn control circadian rhythms and physiological function in nearly every cell of your body. And so this feedback loop is comprised of 4 main circadian clock genes, which I don't need to get into the details for this talk. But what's really important to note with respect to SynGAP especially, is that in addition to this main feedback loop comprised of these 4 circadian clock genes, you also have many signaling pathways or cellular signaling cassettes that can regulate the phasing, the periodicity, the amplitude of this entire timekeeping process. And one of those signaling pathways is called the MAPK cellular signaling pathway. And so if you're not super familiar with signaling pathways, you can kind of think of them as being analogous to, like dominoes, wherein if you think of like a domino effect or like a snowball effect, if you push one domino down, that leads the other one to fall down, and the next one, and the next one, and so on and so forth. Only instead of dominoes, obviously, with respect to signaling pathways, you have one molecule, typically a kinase or one protein, being phosphorylated, and that phosphorylation leads to its activation. And then that then activates the next molecule or the next kinase downstream, and the next kinase, and the next one, and so on and so forth. Until finally, you get the activation of this, effector kinase, or this protein that can then go into the nucleus and lead to a cellular response. So that's kind of, what's happening with respect to this MAPK cellular signaling pathway, where you have a growth factor binding to a receptor on the surface of the cell. And that can lead to the activation of this, first domino, if we're using our analogy. So in our case, with respect to RAS is our first domino in this pathway. And that can lead to the activation of RAF, and then MEK, and then And then can actually translocate or move into the nucleus of the cell where all that action happens. And that can lead to new gene transcription or new, production of new genes. And from a circadian standpoint, this is really, really important because we know that is critical in coupling photic input or light input to SCN clock timing. And so this was first shown in a study performed by Dr. Carl Oberton about 2 decades ago. And what he did is he took mice and he kept one group of mice in total darkness. So he didn't expose them to any light. And then he took another group of mice and he pulsed this other group of mice with light around 9 or 10 p.m. So when the lights were off. And then he pulsed them with light. And he found that the animals he pulsed with light, he saw this really, really massive induction of within the SCN here, which you can see on this bottom image. Again, you can compare that to the control animals at the top that were not pulsed with light. And so what this is showing is that can be activated by a light pulse, especially a light pulse at night, when normally your body is not perceiving light. You're in darkness. And so how this works from a molecular or cellular standpoint is that you have, photic input, photic input that's being perceived through your retina or your eye. And that signal can then be, transferred to or through your brain down to the SCN, that suprachiasmatic nucleus, again, your master clock. And that can lead to this actuation of this domino effect, that cellular signaling pathway that we just mentioned. And once is activated, remember our kind of effector kinase in this case, it can lead to the activation of downstream proteins that can go into the nucleus and lead to new gene transcription. And this is really important because some of these new genes that are generated are actually those core circadian clock genes, especially this period 1, for example. And once that happens, once these new genes are produced or transcribed, that can actually allow your body to entrain to this new light that it's being perceived. And so this is kind of, again, what's happening at a cellular level. Your body is perceiving this light, and you have this MAPK pathway that's helping to transduce or translate that light signal into something that your body can actually use to adjust its phasing to the environment. And so how does SynGAP play a role here? How does it fit in with ERK? Well, SynGAP is one of the most abundant proteins that sits at this postsynaptic density. So you can see it right here in yellow. And it functions as a negative regulator of RAS. So again, if we're recalling, kind of our domino effect, you see that SynGAP is sitting here and it's negatively regulating the expression of RAS. And this can then go, this signaling pathway then goes RAS to RAF to MEK and to ERK. And so again, this is just showing that SynGAP functions as a negative regulator of this pathway, that's so important to circadian clock timing. So, knowing that, SynGAP is involved in regulating synaptic plasticity, and that the circadian, or that the ERK MAPK cellular signaling pathway is so crucial to coupling light to this, clock timing within the brain, that begs the question, is SynGAP actually expressed in the SCN? And if so, could it be a regulator of circadian clock timing? So that's kind of what we're getting into with this paper that I'm going to talk about. And so we first wanted to see, again, whether or not SynGAP, at least SynGAP protein, was expressed within this master clock region. And so you can see here, I immunolabeled this mouse brain section. So this is just coming from, I hate using the word normal, but this is coming from a wild type animal. So it's not, genetically modified in any way. This is just a wild type mouse. And I stained this mouse brain section with a SynGAP antibody. And you can see here that when we kind of zoom in to the SCN, you can see this nice, really robust expression of this SynGAP protein within the suprachiasmatic nucleus. And here's kind of just a prettier picture. So this is an immunofluorescent labeled mouse brain section, where SynGAP you can see is in green, and then you have NuN, and NuN is a marker of neurons in red here. And so this is just showing that SynGAP is actually expressed in neurons, which is exactly what we would expect since SynGAP is, sitting at the synapse. And then finally on the right side here, these are just associated cells from the suprachiasmatic nucleus. So instead of, me staining this mouse brain section, here we're actually staining dissociated or individual cells within this SCN region. So now we know that, SynGAP is indeed expressed in the SCN. And so now we wanted to ask the question, is its expression in the SCN rhythmic or clock regulated? And what I mean by that is that many genes throughout the body, they change, their expression level changes across the day. And that can be a really key indicator as to whether or not the circadian clock is actually controlling the expression of SynGAP and of other proteins or other genes. And so what I did to kind of answer this question is I took two groups of mice. And so the first group of animals, you can see here on the left side, was sacrificed during the circadian day time point, so around noon, 11 a.m. or noon. And the second group of mice was sacrificed during the circadian nighttime point, so around 8 or 9 p.m. And then what I did is I took the SCN, or I dissected out the SCN from these animals at these two different time points. And I kind of mashed it up in a way and collected this homogenate. And then I ran this, this protein lysate on what's called a Western blot. And Western blots allow you to visualize the expression of proteins on these membranes here, which you'll see in gray. And so when I did this, you can see here that on the right, the animals that were sacrificed, during the nighttime, the bands here, these Western blot bands, are much darker than the ones on the left. And so what that's suggesting is that SynGAP is more highly expressed during the circadian nighttime compared to the circadian day. And you can see that here in my quantification as well. And then in order to get kind of a more detailed readout as to exactly, when SynGAP is most highly expressed, what we did now is we took 6 different groups of animals and we sacrificed these animals at these 6 different time points throughout the day. And so here CT is referring to circadian time. And so CT2 would be 2 hours after the lights come on. And so typically mice, kind of like humans, mice are housed in a standard 12-hour light, 12-hour dark schedule. So their lights will come on at about 6 a.m. and they'll go off at about 6 p.m. And so CT2 would be 8 a.m. So it would be 2 hours after the lights come on, or should come on. And so what we did is we sacrificed these different groups of animals at these different time points. And then I labeled these brain sections, so these SCN sections coming from these mice, with the SynGAP antibody. And I found that SynGAP was most highly expressed during the middle of the circadian night, or the CT18 time point, which you can see here. So now we know that SynGAP is expressed in a circadian manner, but in order to confirm this, what we did is we took BMAL1 null animals. So now instead of being these wild type mice that don't have any genetic mutations, we took these BMAL1 null animals that lack this core circadian clock gene BMAL1. And because they lack this core circadian clock gene, they have no, or their circadian system is non-functional, right? And so when we sacked these animals, during the circadian day, so at CT6, circadian time 6, and circadian time 15, so this would be the nighttime point, and immunolabeled these brain sections in the SCN with this SynGAP antibody, we found that there was no significant difference between the day and the nighttime point. And that's exactly what we would expect, right? Because these animals don't have a functional circadian system. And if SynGAP is regulated by the circadian clock, then we wouldn't expect SynGAP to show different expression levels, at different times of the day. And so these data confirmed that SynGAP is indeed regulated by the circadian clock. And now we wanted to look at phospho-SynGAP expression in the SCN. And the reason why we wanted to look at phospho-SynGAP expression is because this active form of SynGAP has been shown to play a really important role in its functionality, especially with respect to synaptic plasticity. And so what we did is exactly what we did with that total protein. So we took 6 different groups of animals and we sacrificed them at 6 different times of the day in order to see whether this expression is going to change across the day. And what we found was a really interesting, expression pattern, wherein in the SCN core, which you'll see here in blue, let me backtrack just a bit. There's 2 main subcompartments or subregions of the SCN. The first being this retinorecipient is what it's termed, SCN core in blue, which is kind of that smack dab center of this teardrop shaped SCN structure. And then you also have the SCN shell, which kind of just sits right above the SCN core. And what we saw was this really interesting phenomenon wherein phospho-SynGAP or active SynGAP expression was most, was the highest in the circadian night within the SCN core, so at this CT18 time point. But if you look in the shell, again, this red part right here, we found that its highest expression was during the daytime. And so that was, again, was a pretty interesting find because that kind of follows, calcium signaling, the expression pattern of calcium within the SCN. And it also goes to show, again, that, phospho-SynGAP within the SCN is rhythmic. And then finally, we wanted to see whether SynGAP expression was induced by light. And so what we did is we took animals and we took one set of animals and did not expose them to light. And we took a second set of animals and exposed them to light. And you can see here that the animals we exposed to light and then, we sacrificed them and labeled their SCN, their brain tissue, with this phospho-SynGAP antibody, we found that they actually displayed an increase in this phospho-SynGAP expression. And this confirms that SynGAP is indeed induced by a light pulse, at least during the circadian nighttime point. So again, around 10 p.m. when the lights should normally be off. And so given that SynGAP is expressed in the SCN, and it's regulated by the clock, we then asked, could its dysfunction lead to alterations in circadian clock timing? And so this is where the SynGAP heterozygous mice came in. And so we obtained these SynGAP het mice. Dr. Gavin Roomba from Scripps Institute was very generous and provided us with his het mice. And that allowed us, to ask or to examine at the functional level, when SynGAP expression is altered, could that then contribute to deficits or any sort of abnormality in circadian clock timing? And so that's exactly where we started. And so what we did is typically a great way to assay or to determine whether or not circadian clock timing is altered or whether it's intact is to put mice on running wheels. And so essentially how we do this is we get these really big, typically they're like big rat cages, and they're equipped with these running wheels, and mice are allowed to run, whenever they want to. They're free to run, whenever they'd like. And then what we do is we put them on these wheels, and mice are nocturnal, right? So they're going to typically run during the nighttime period. And so this nighttime period is denoted by these gray bars here, and the daytime period is denoted right here by these like white lines essentially. And so what we're able to gather from this is that each one of these black tick marks, I know it's a little bit difficult to see, but each one of these black tick marks is indicative of a 5-minute bout of activity. And so again, because these mice are nocturnal, they're going to run during the dark phase here. But what's interesting is when you compare the wild type mice here on top to the SynGAP het mice here on the bottom, you can see that these SynGAP het animals display more overall activity during this dark period. You can see that here by our quantification as well. And then here again is kind of recapitulating what I just showed, but this is considered like an activity plot of this wheel running behavior. And so you'll see Zeitgeber time 0 through 12 is referring to that period typically around 6 in the morning to 6 at night when the lights are on. And then Zeitgeber time 12 to 24 is referring to the time in which the lights are off. So the overnight period, 6 p.m. to 6 a.m. the next morning, that dark period. And so again, you can just see that these SynGAP animals here in red display an increased or a heightened level of average activity during this dark period relative to these wild type mice here in black. And then in addition to examining wheel running behavior in the standard light dark conditions, so that 12 hours of light and 12 hours of dark, another way to actually look at the endogenous free running rhythms of animals is by putting them in constant darkness. And so the reason why we do this, and I don't think I mentioned this earlier, but circadian rhythms, they're endogenous, right? So they can function even in the absence of external timing cues. So even if you're in constant dark conditions and you're not receiving light input from the sun or from your external environment, your circadian system can still function. And so when you put these animals or these mice in constant dark conditions, which you can see here on the bottom of this top graph, it's denoted by this, the all gray bars. When you do this, this allows you to determine the circadian period or tau of the animal, which is typically between 23 and 24 hours, which you'll see here. And so when we did this with SynGAP, with wild type and SynGAP animals, we noted that there was no difference in the circadian period when they were, put in these free running conditions, when they were not exposed to any light. But what we did observe was that during this transition period, or during the time, it's known as the offset. So when they stopped running to the time when they started running again the next day, you can see that they have this increase in activity. And you can kind of tell that by looking again at those black tick marks here. If you can compare this, the black tick marks here in the bottom graph to the black tick marks here in the top wild type graph. And then there's our quantification as well. Additionally, you can also kind of test light sensitivity as well in these mice by exposing them to constant lighting conditions. And you can also get a readout of their period in these constant light conditions. So now, instead of putting these mice in total darkness, what we're doing is keeping them in total, or we're constantly keeping them in dim lighting conditions. And so you can see here, dim light typically causes wild type animals to display less overall activity, which you can note here. It kind of becomes a little bit scrambled, and these mice just don't run as much. But you can see in the SynGAP mice, they display more overall activity compared to these wild type mice on top. Interestingly, we also noted that the period, so this tau, is increased in these SynGAP het animals compared to the wild type mice. And so when you put an animal in constant light conditions, instead of the tau or that period being 23 and a half or 24 hours, the period's actually going to lengthen. So it'll be between 24 and 26 hours, which is exactly what we're seeing. But again, these SynGAP het animals seem to have an increase in this, or sorry, a lengthened tau, a potentiated period compared to these wild type animals, which was also quite an interesting find. And so finally, we wanted to examine light-evoked ERK activation and behavioral phase shifting capacity. And what I mean by phase shift is that essentially that means that you're moving your body's internal time either earlier, that would be like a phase advance, or later, like a phase delay in the day. So you're kind of moving your wake-up time or your bedtime earlier or later in the day. And so you can see here a great way to test not phase shifting capacity necessarily, but a great way to test light sensitivity or light responsivity in animals is by looking at ERK activation after a nighttime light pulse. And so again, I mentioned this earlier when I brought up Dr. Oberton's study showing that ERK is indeed induced by a light pulse. And so we did this paradigm again. And so how we did this is we took wild type mice, And we left one group of wild type mice in constant dark conditions, so we did not expose them to light. And we took another group of wild type animals and exposed them to light. And you can see here, just like I mentioned with that study about 20 years ago, in the mice that were exposed to light at night, they displayed this really rapid, robust increase in phospho-ERK expression within the SCN. But when we did this same thing in SynGAP het animals, which you can see here on the right, they actually displayed a potentiation in this ERK activation. And you can see that because it's much more dark, right? This expression of phospho-ERK is much darker in the SCN. And then again, you can tell that with our quantification as well. And so that's kind of a more, cellular or molecular readout of the ability of an animal to respond to light. But then to examine phase shifting capacity from a behavioral level, what you can do is you can actually put these animals back on wheels and leave them in constant darkness. So you just allow them to run, and they're not being exposed to any light for a period of 2 to 3 weeks. And then after this 2 to 3 week period, what you can do is you can actually pulse these animals with light during the circadian nighttime point. So here we pulse these animals with light at CT 15, circadian time 15. So that would be 3 hours after they, 3 hours after they started running. So right here, you can see, I don't know if you can see that yellow arrow. So that's where we pulse these animals with light. And then what happens is when, in wild type animals at least, when you pulse animals with light during the early subjective night, so CT 15, what happens is that the next day, these animals will start running later at night. So for example, if on this day, right, if before we pulse the animals with light at night, if they started running at 6 p.m., for example, if you pulse those animals with light, the next day they might start running at 7 p.m. And so that's a great way to assay, phase shifting capacity within these mice. And so when we did this in wild type and het mice, we noticed that these het mice displayed a potentiated phase shift compared to the wild type animals. Which again just suggested that these het mice show not only increased light-evoked ERK activation, but also an increase in the behavioral phase shifting capacity. And so that's kind of the gist of the study from our paper. And so again, the key conclusions that we found is that in wild type animals, the SynGAP or SynGAP is expressed within that SCN, right, within that master clock region. We also found that total SynGAP expression and also that active form or that phospho-SynGAP expression within the SCN is rhythmic, and that SynGAP can be induced by light. And then again, we looked from a more functional perspective using those heterozygous mice and found that these het mice display alterations in circadian wheel running activity, and that they also display enhanced light-evoked activation, ERK activation, and behavioral phase shifting capacity. And so these are kind of the key findings from our study. And so I don't want to get too much into this, but I have already gone through the pathway a bit. So hopefully it'll be a little bit easier to understand now. But essentially what we think is happening, or we're postulating now what could be the cause of this increased light sensitivity in het mice, right? So why are they displaying this increase in ERK activation or this potentiated phase shifting capacity? What's happening from a cellular level? Well, when the eye perceives light, that light signal again is going to travel down the brain. And it's going to activate this, if we go back to our cellular signaling pathway, right? Like our dominoes. So it's going to activate RAS and then RAF and MEK and ERK. And then ERK then can translocate to the nucleus and can lead to the, in the nucleus, new circadian clock genes can be generated to allow your body to reset to that light pulse. But what's happening potentially is that in SynGAP het animals, SynGAP het animals are losing this ability to negatively regulate, this ERK MAPK cellular signaling pathway. So you have less of this negative regulation of ERK. And because of that, potentially what you could see is you could have these more and more AMPA receptors being brought to the surface, leading to an increase in the excitatory drive. And potentially that could be explaining why, we're seeing this increased phase shifting capacity. And so I kind of wanted to, again, I'm a basic scientist. I'm not a translational or clinical scientist, but I do think it's kind of interesting to put this, what I've shown in more of a I guess, clinical context as to why we're seeing, this potentiated phase shifting capacity to light, and what could that mean? And so again, we've known for quite some time that light is the most important time cue in the body, in the ability for our SCN, that master clock, to regulate rhythms and to reset our body clock to the light input that we're perceiving. And so I think we've all, potentially heard, or most of us have heard that, light at night, like it's bad for your health. And the reason why it's bad for your health is because we normally shouldn't be perceiving light at night, right? So melatonin, one of the key hormones that allows our body to go to sleep or leads to sleep, can actually be repressed by light. So normally melatonin, it is not released or produced during the day. It's produced at night when the lights are off. But when our bodies are perceiving these blue light signals, whether it be from our iPhone, our tablets, any light timing cue, that can actually, alter melatonin rhythms and make it more and more difficult for you to fall asleep. And so essentially, we did not test this by any means, but if these het mice from our study, are showing this potentiation and this phase shifting capacity to light, that could also mean that these, kids with SynGAP, are even more so affected by light at inopportune times compared to, normal kids, for example. And so the fact that these, if kids are perceiving light at night, that could further exacerbate these sleep issues that they're experiencing. And so that's kind of one functional take, perhaps, from this study. And I also wanted to kind of at least bring up a few other considerations. Again, this is nothing that we went over in our study, but I think it's really interesting too to note that we used Dr. Gavin Rumbaugh's heterozygous mice, and these heterozygous mice seem to be a really great translatable animal model for the study of sleep and circadian dysfunction, not only based on our study, but also based on several studies from his lab and other labs. And what we could actually do, and again, we did not do this in our current experimental model, but we could actually cross or breed these heterozygous mice to another mouse line in order to potentially rescue SynGAP expression levels upon the administration of a drug called tamoxifen. And that would be really, really an interesting study to perform, because we could see if we could, perhaps rescue some of these circadian abnormalities, if you will, in these adult animals, right? So these animals would be past the developmental stage at this point. So it would be interesting to see if rescue of SynGAP protein levels can also rescue or lead to more normal circadian activity in these mice. And then finally, I also think it's important to note that the circadian or your circadian clock, your SCN, controls more than just the sleep-wake cycle, right? So that's just what we think of when we think of the circadian cycle. We think of sleep because of that 24-hour process. But the circadian clock actually regulates so many other processes, from cognition, metabolism, glucose homeostasis, hormone secretion, etc. And actually, cortisol and melatonin are really key biomarkers for the health of an individual, right? And so it's a great marker to tell, whether a drug is functioning correctly, whether your immune system is functioning correctly. And so I think that's also just something great to consider. And that kind of brings me to my next point of this, chronomedicine and chronotherapy. And that, I don't know how to explain this the best way, but this is kind of a hot topic right now in the circadian field, because it's showing that it's taking into account the fact that your circadian rhythms are so important, because oftentimes our internal rhythms, right, our body clock is actually different than the external time around us, than the actual clock time, for example. And so this is a hot topic right now in the more clinical circadian side, because if we're actually taking into account the fact that, we each have our own individual rhythms, that could be really crucial for when we administer drugs, for example, because that could make, drugs and other medicines or therapies more or less efficacious if they're administered at different times of the day. And so it's kind of getting into this more personalized medicine, which I think is really, really cool. And I think it gives a lot of hope, especially for, SynGAP kids and other many other neurodevelopmental disorders, or children who have these neurodevelopmental disorders that often have sleep comorbidities. Because I think, you know, once society starts to realize, hey, it's really important to actually consider your body's internal clock, you know, when we're administering these medical therapies, I think that could also, you know, prove to be really, really efficacious in treating some of the other, you know, comorbidities that you're seeing in SYNGAP kids. So I just thought that that would be really nice to note. I think it's something that's really up and coming and really exciting. Again, it's a bit off topic, but it's just important to note that, you know, there are scientists, physicians, clinicians alike are definitely starting to take into account, or into consideration at least, the fact that, you know, examining our internal time is very important, and it can vary, you know, from individual to individual. You know, if we think of morning larks versus night owls, is a great example of that. So I think that's a great consideration as well. And then finally, I just want to thank my lab, so Dr. Carl Oberton, for allowing me to do all of this research, in addition to Dr. Carrie Hoyt, Dr. Jun Yoon, Anisha Kaladindi, also Dr. Gavin Roomba for providing his SynGAP mice. Dr. Huganier provided us with some of the SynGAP antibodies, in addition to Dr. Araki. And then Monica Weldon from Bridge the Gap also helped to distribute some of those circadian and sleep surveys to allow me to get more of that, you know, patient data, which was really exciting. So yeah, I'm happy to hopefully answer any questions that you might have. Should I end my slideshow now? Is that? Oops.
Right. Yeah, you can go ahead and end the slideshow.
Okay.
Thank you so much. This was very, very good. We really appreciate it. I think Hans has some questions.
Yeah.
And Hans, go ahead.
Yeah, can you guys hear me okay?
Yes.
Yeah.
Super. Sydney, first off, congratulations on successful defense of your thesis. I actually spent a couple of summers looking at serotonin in the SDN and the LGL in an anophthalmic mouse. So it was really a trip down memory lane.
Yeah.
But I'm wondering, you know, mice are easy. You can measure their activity on a running wheel. Is there anything wearable or anything modern that lets us look at our own children? I mean, is that, you know, an Apple Watch function nowadays that we should just strap on them?
Definitely, yeah. So currently in the clinic, kind of the gold standard right now is looking at what's called dim light melatonin onset. And so that's where melatonin, usually melatonin is taken from either the blood, urine, and even saliva nowadays. But it can be measured every 30 minutes to an hour, especially during that day to evening transition when melatonin is typically produced. So that's kind of the gold standard. But again, you have to be in clinic for that, right? So it's not as efficacious, I guess, or that's the gold standard, but it's a little bit harder to do unless, you know, your child is in the clinic for days and days. But yes, there are actually more wearable devices, circadian wearable devices that allow you to track like body temperature, because that is actually regulated by the circadian cycle. And I don't know what other, I don't know exactly what other specific, you know, outputs are on these wearable devices, but essentially exactly like what you said, they're like watches, for example, and that's something that's really, really exciting. Actually, Dr. John Hoganesh at Cincinnati Children's Hospital is kind of spearheading some of this new chronomedicine, which is really, really exciting. But yeah, there are definitely wearable devices. I don't know that there are any that are, you'd have to ask the physicians. I don't know how, you know, publicly available they are just to buy off of Amazon. But yeah, definitely measuring cortisol and melatonin, and then these wearable devices, which again are kind of the hot up and coming, you know, technology for circadian or chronomedicine.
Quick question. Sorry, Hans. I was just going to ask, just based on that, if our kids are getting overnight EEGs or if they're already getting sleep studies, is that something that we could be potentially pulling at that time? And
definitely, yeah, that's
days of data, or is like one night's worth or 2 nights worth enough?
Yeah, definitely. That's more. I think typically it's like a 2 to 3 day period. But if you're, you know, in the clinic for a night, certainly getting, you know, whatever especially if you're measuring at a, you know, semi-regular interval, so every hour, for example, especially during that transition phase, like I mentioned, is perfect. Yeah. And again, I'm not a clinician, so you'd have to ask your doctor, your physician, if they would be able to do that or willing to do that. But it's something great to consider for sure.
because you emphasize the MAPK pathway, you know, a few parents, a few, you know, case reports describe statin therapy for these kids and its effect on the MAPK pathway. Is that something that's been identified where, let's say, their, you know, run wheel activity or some other behavior can be a biomarker, you know, where you can give statins in different doses and potentially see different results in a circadian output?
Oh, that's a great question. Yeah, so if you did it in mice, definitely you could put the mice on running wheels and then just, you know, examine their wheel running activity, either at baseline or in those constant dark, you know, free running conditions to look to see if their tau is altered, or whether that affects, again, their phase shifting capacity, that, you know, the ability of their body to align with this external light, whatever you make that external light. If you want to, you know, push it and change it so it's not a standard 12 light schedule, you could certainly do that. So from mice, I think that would be incredible. That would be a great study to perform. I don't necessarily know. Are you asking from more of like from a clinical standpoint? So for, or?
Yeah, statins are pretty controversial. They've been tried before in like neurofibromatosis. But if you could show, you know what I mean, let's say a dose effect on the MAPK pathway in a circadian mouse model, I think it would be, you know what I mean? It would show some physiologic credence to the effect on that pathway.
Definitely, yeah. And I would say definitely the wheel running behavior would be the first assay of choice, and then also just examining that, you know, ERK activation after you give statin, or put that, exactly, yep, or couple that with a light pulse as well. If you look at the dose response curve and couple that with, you know, light-inducible activation as well, to see if light potentiates that effect that you see with dose. Yeah, I think
Great.
that would be really, really interesting. But
you'll get us that protocol.
Yeah, definitely.
And then,
oh, go ahead.
you know, I just have one last question. You know, do you happen to know, embryologically speaking, are these SCN neurons like differentiated very early in brain development, or are they sort of downstream and a little bit later?
That's a great question. So your circadian, you still have rhythm. Cells in your SCN are rhythmic right after birth, but I'm not sure about, you know, I don't know enough about the development of the circadian system as far as, you know, P0 mice compared to P10 compared to P21. I can tell you that all of our studies that I've done are always in adult animals, but I'm sure there is some sort of developmental effect. But like I was mentioning, I think, I don't know if you all have watched Dr. Roombaugh's or followed Dr. Roombaugh's work, but I think this Cree rescue mice is really, really exciting. Yeah, just to see if you know, restoration of SynGAP levels after this developmental period could also rescue some of these circadian phenotypes. I think his lab has shown it before. It rescues some of those cognitive deficits, and I think some of the markers of epilepsy as well. So I think that would be a really interesting study for sure.
Super, thanks so much. What are you planning on doing? You know, are you sticking around at your institution or going elsewhere, or?
No, yeah, I'm moving to Boston, so I'm actually studying. So, like I mentioned, I'm not a sleep scientist, but I'm still studying circadian rhythms, and I'm also dabbling a lot more into sleep, which I'm really excited about. So, yep, postdoc for sleep and circadian rhythms. Yeah.
Where's that happening?
Harvard. So, yeah. So, I'm very nervous. That's the goal, but we'll see.
And who's going to be your mentor?
Dr. Clifford Saper. And I'm also potentially being co-mentored by Dr. Stephen Lockley, which is really exciting too. So they're both seem awesome.
Okay.
That's great. Congratulations.
Thank you so much. Thanks for the questions. You brought up really great things to consider for sure.
Then I have a question. Looking at your graphic, seems to be that the SynGAP mice is always hyper compared to there is any time that you saw the reverse? The SynGAP mice more mellow?
Never, you know, I've never seen that. And it's funny that you asked that. And I think actually Dr. Kadam mentioned that in her webinar, and also she has a recently published paper that shows that as well. But I could actually phenotype the mice So typically we snip the tails from the mice to genotype them. So we run a PCR to see if they're actually hets or wild type. But I could actually tell which animals were hets just, from the day they were, from the time they were 21 or 22 days old, just because they were so hyperactive in their cages. And that was, so I would compare and I would note and kind of mark their ears, and then I would genotype later and compare to see, if I was correct. And so, yeah, I never saw the opposite. I always saw that they were hyperactive. But that would be interesting. I mean, I didn't sit there, right, and observe them for 24 hours straight. So I guess I can't tell you it. I don't enjoy being at the lab at 2 a.m. So I wasn't just staring at them, seeing if they were maybe less hyperactive at certain times of the day. But every time I interacted with these animals, they did show this hyperactivity phenotype.
Okay, and then melatonin is quite popular in SynGAP patients. A lot of parents will use it. Any comments about using over-the-counter melatonin?
Yeah, so I'm not a physician. I can say that I think it's, from what I've read, it's not great to take, over 5 milligram, a 5 milligram dose over the counter. And also from what I've heard, and again, I think it's kind of, I don't want to say there's discrepant information, but I think if possible, it would be great for melatonin to just be temporary, because your body already produces melatonin, right? So essentially it should be, it's a hormone that's naturally produced. And so if potentially you could wean yourself off it, I think that's kind of the best option. But certainly I've taken melatonin for days and weeks that I can't sleep. And I think it works great. But yeah, I'm not a physician. I can't answer that question too well, but.
And then you mentioned the blockage of the blue light.
Mm-hmm.
Do you think the glasses now that they sell so easily that you can block the blue light, it will be helpful for SynGAP patients?
Yeah, for sure. Yeah, I actually didn't know that they had glasses. I know, so like on my phone, I know there's like a red light setting. So that's typically what I'll do 2 or 3 hours before my bedtime. I'll set my phone to that red light setting, but I didn't know there were glasses. That's really, really cool. But yeah, definitely anything that can, block or prevent that blue light, because blue light is what's activating your melanopsin expressing retinal ganglion cells. Red light doesn't do that. So red light isn't activating that, retinal hypothalamic tract that's activating these SDNs. And so, or the SCN. And so that's why, these glasses, like you mentioned, which is so neat. I haven't heard of that. Would be so beneficial, right? Because you're not exposing the child to that blue light. And so potentially that could then keep your melatonin rhythm at bay a little bit more. So definitely, if you can keep them, if you can make them wear the glasses at night. I don't know how comfortable they would be trying to sleep with glasses on, but maybe, but.
Okay, and then I think, Peter, we have 2 questions on the.
Yeah, I was going to ask just one question first, if that's all right. So I really appreciated this talk. My son woke up at 4:30 this morning, so it's very relevant to us. We find that giving actually like a small dose of melatonin tends to be pretty effective, and like just like 0.5 milligrams even.
Yeah.
But then I think what a lot of SynGAP families see is that helps the children fall asleep, but it's really the problem of keeping them asleep.
Staying asleep,
and waking up, early in the morning, for instance. What's like the melatonin, what's the significance of melatonin in the different phases of sleep, I guess, from falling asleep to staying asleep, and I guess early morning type hours as well of the cycle?
Yeah, that's a great question. So I don't know the answer to that particularly. I know it's continued to release throughout the night until that, circadian drive for arousal. So that's when your norepinephrine is going to kind of take over and tell your body to wake up, around early in the morning, 6 to 9 in the morning, I guess, depending on if you're a morning lark or a night owl. Yeah, I'm not sure about the actual, kind of kinetics of melatonin, so to speak. But yeah, I know that's a huge issue, is actually getting children to stay asleep. They might fall asleep, but then wake right back up. And have they ever been, diagnosed with like a formal sleep disorder, like an irregular sleep wake or anything like that? Or
No. No, we haven't had like an actual sleep study done on my son in particular.
Gotcha.
So we haven't had any sort of formal diagnosis.
Right.
You know, we do give melatonin, and then we're also prescribed THC from a neurologist as well for sleep.
Right.
So that has been basically kind of the most effective pairing for ourselves.
Okay. Yeah. Yeah. I'm sorry. I'm not sure about exactly when, you know, melatonin or how long. Yeah. I can't tell you the answer to that, but.
And I think JR has a few questions here. She is a SynGAP parent and a geneticist. I'm going to go ahead and just unmute her so that she can just go ahead and ask her questions. JR?
Hey, yeah, hi. Thank you so much, Sydney. So I'll just read these questions. First off, this is such amazing work. I love what you did. My first question is really, what would you most like from the SynGAP community in order to continue your research? And so, like, one question and sort of where I'm getting to is, are you interested in studying other variants other than just the straight het?
Oh, that's a good question. So, are you, by other variants, do you mean like look at like a? I'm sorry.
Missense.
Oh, I see. You know, I haven't even thought about that. That would be really, really interesting for sure. And I know I was talking to Marta earlier before the talk. I'm really interested in kind of dabbling onto that clinical side. And so I did a bit with that Bridge the Gap study that I was mentioning. So that's kind of my passion with respect to SynGAP now, is kind of trying to correlate what I'm seeing at the molecular and cellular level to, you know, these kids who have sleep issues, kind of dabbling more into that. And I know, like I said, Dr. Kadam and I think Dr. Holder are doing some of that. But I have not thought of looking at you know, missense mutations rather than the standard, you know, pathogenic variants. That would be very, very interesting. And I can tell you that we didn't do anything specifically with the, so basically these SynGAP het animals that we got from Dr. Roomba, they do actually survive, the homozygous mice survive to like somewhere between P3 and P6 or 7 is what I was seeing. So about a week, they don't live past a week. So I actually think it would be kind of interesting to see whether, you know, we see any circadian gene changes or like that seem super, you know, abnormal or different than the wild type. Again, that's difficult because you can't put week-old mice on running wheels, right? So you can't assay from a behavioral perspective what's happening in these homozygous mice. But I do also think that would,
stuff.
Yeah, exactly. So we couldn't do yeah, perhaps definitely, but we couldn't do anything behavioral with that wheel running assay. But what we could do is use like a cell culture model, or even cross, you know, that SynGAP mouse with a you know, a clock gene reporter mouse to actually look at clock genes, you know, in vivo, you know, in a live SCN section, for example. So we can keep that alive and then monitor in real time, you know, clock gene expression, you know, using a confocal microscope, for example, over days at a time. Because the SCN, it's really, really cool because it's so hardy, right? So the SCN is comprised of a lot of GABAergic neurons instead of these, you know, excitatory glutamatergic neurons. So it actually can survive, you know, quite some time in culture. So I think that would also be really, really cool to see if, you know, we see an even more profound effect in these homozygous animals, which I would expect. But definitely looking at other variants would be very, very cool. Again, all I've thought of right now is kind of going to the clinical, more clinical side, but yeah, I think that would be very interesting.
Great. And so I assume you know about our citizen study, right? That has over 100 patients and all the genetics and all the electronic health records. Yeah. So that should be great to look at.
For sure. Yeah. Marta and Mike and I were talking about that earlier. And I think they mentioned that it had the genetic reports like you mentioned too.
Yes,
they have all the information that the parents provide essentially, which is so helpful for sure.
Right. So we should have pretty good. Yeah.
Exactly.
Sorry, we should have pretty good sleep info too in there.
Definitely. Yep. And I can start to look at like patterns between, these different variants, etc. So yeah, that would be incredible.
So I realize that you're not a clinician right now, so I don't want to put you on the spot too much, but as a parent, you know, I'm trying to understand a little bit of what I can change and what I need to work with and accept in my kids' sleep, right? So looking at the het behavior in the mice, every slide you put up was just beautiful. And I kind of understood it in the moment, but it went by really fast and I don't really think about this much. So I don't have a good synthesis in my mind of what you said about the hets, other than that the lack of SynGAP definitely changes things in the SCN. So do you have a sense as far as like phase shift, like I know there's some people that maybe have like a 25 or 26 hour clock, right? They don't have a 24-hour clock. So I know that happens in humans. Do you see anything for the mice? Like, is it better to try to shift them a little, or are they just, I mean, to me, they just looked more active, like a lot. They looked more active in the day and the night, actually.
Right. So better to shift them as in?
Well, like, yeah, like, so like, I think for our pediatricians always say, you know, for sleep entrainment, like do the same thing every night and, you know, I'm sure the chrono medicine, I forget what you called it, but
yeah.
You know, it's going to be like reduce light levels as you're getting towards bedtime, probably do the same thing at bedtime every night, right? Like this is called sleep hygiene, maybe like trying to get, you know, and like sleep hygiene never worked at all, right? Like like what my child has is like way beyond sleep hygiene. So I was wondering, do you think it's better to, or not, do you think it's better? Because I know you're not a clinician, but in the mice, did you notice anything like, do you think the mice would have done better if you just changed their clock a little, or if you
I see. Yeah, that's a great question. And so we didn't do anything where we like, I don't know how to phrase this in a, so where we like pushed the system, because like I was mentioning, I can't remember whose question I was answering earlier, but you can do different light manipulations. So we could do what's called like a T7 light cycle, or or put the mice in a light cycle that's not standard. So not a 12-hour light, 12-hour dark schedule. And that can kind of push the system, you know, just to see how plastic the circadian system is essentially. And so we did not do that with any of these het mice. That would certainly be interesting, you know, to see whether these het animals would be able to entrain better. And I'm sorry, I probably should have explained this better. When I say entrain, kind of what I'm saying is that your body, so your internal or your innate clock, is able to have a kind of a phase relationship with the actual, you know, clock time. So whether your body is able to kind of, I don't know how to understand the external clock time. And so that's something that we did not do with these SynGAP mice. But you're right. I kind of understand what you're saying. I think that would be interesting, you know, to see if we would have done different light manipulation paradigms to see how they would have, you know, acted, or to see what the output would have been, the behavioral output at least.
Yeah, because I'm wondering if in order to get better sleep, like maybe would something crazy like going to bed, trying to go to bed at 8 and then being awake from midnight to 2 and then going to bed again or something. I mean, I don't know. I'm not saying that is like what I want. I'm just wondering if there's something that would work better. So like sometimes for my SynGAPian, like changing the environment for him is better than trying to change him. So I'm trying to figure out what is it, if anything comes up of what might be the best sleep schedule.
Definitely. Yeah. And I can't answer too much of that because I'm not a clinician. So typically what people will do that have like delayed sleep phase or syndrome, for example, where they need their body to be advanced, right? If they can't go to bed until 2 in the morning, then what you can do is you can give melatonin at night, you know, earlier in the night to help them fall asleep, or you can try to phase advance them a bit to make them more like the actual clock time. By giving or pulsing them, you know, with light in the early morning to try to advance their schedule. But again, it's not exactly like what you're saying, because it seems like your child is experiencing, you know, wake up and fall asleep, wake up and fall asleep. So I'm not exactly sure what you would do in that case. Yeah.
So like using the high intensity lamp, like for seasonal affective disorder. I wonder if doing that in the morning would be helpful potentially.
Yeah, perhaps. Yeah. Again, I know that's a great therapy for individuals who have an already delayed phase, but I'm not sure if it's you know, kind of irregular. Yeah, it sounds like yours is more irregular. So I'm not sure how beneficial that would be, but you could certainly ask, you know, your clinician.
Yeah, because my SynGAPian's need for sleep doesn't seem to change. Like he could wake up at 12:30, you know, right just after midnight and be awake all day, or he could wake up at 6 and be awake all day, or he could sleep till 7 and be awake all day. He doesn't seem to get more tired.
That's so interesting, huh? Yeah,
Super interesting. Yeah.
I know it's probably not much fun for you. Yeah, very interesting, but
well, it has also gotten better as he's gotten older. So he's quite old now, so it's better. But okay, I had another question though. Oh yeah, my guy seems to see in the dark really, really well. I was wondering if you had any idea on that, or if that's just retina and doesn't have anything to do with anything else.
That's a great question. No, I don't know. I just like better than you, like he can see
way better than me.
Oh,
yeah.
Interesting. Yeah, I have
even when I was younger, he could see way better than me. So
is it just like shapes, or is it more like he can
walk around in a dark room and not hit anything? And his brothers couldn't, you know, his same age brothers couldn't.
Right.
He seemed to have way better vision than the rest of us in the dark.
Yeah. I mean, I think that's more of a, like you said, kind of like an eye thing, but that's very, very interesting. I've never heard that.
We've heard the same thing from a lot of parents that their children.
Really?
I have the same case. Yeah.
Where night vision seems to be just like really, really like very, very good, very precise.
Better than average. Yeah.
Yeah, we'd be like in a messy playroom in complete darkness, and he could walk around and not hit anything, whereas I couldn't walk around.
You cannot sneak around them in dark to try to get something, because they just detect you right away. They see everything.
Very interesting. Yeah, I don't know. I can't answer to that. That's very And what about during like in so in the daylight, so in light conditions?
Sensitive.
Do you see like better sensitive? Okay.
Sensitive to light, yeah. Right. Like needs a hat on a lot. Yeah.
Okay. Yeah, I'm not sure. I think that would be more of an eye, a visual or retinal thing.
I just wasn't sure what role ERK was playing in there. Maybe some, maybe not. Yeah. Okay, I want to switch back to research. There are other models for sleep, sometimes like fish. I don't know if worms have a model, organoids. Do you think there's any, do you follow any of that research for circadian rhythm? And do you think this could be useful to look at SynGAP in these, or is that just too far afield to ask you that question?
No, that's a great question. So I don't so much. I mostly follow mammals, so mice. There's not quite as much in rats, but mostly mice. Drosophila is also a really great model for circadian. That's kind of the Drosophila and mice are kind of the two big ones. But yeah, definitely it could be used in other models. Again, I just follow mice, so that's what I know. But organoids. So by organoids, do you mean like looking at, so kind of trying to look at like whether or not certain drugs are more or less? I guess what do you mean by organoids? Just looking at.
I don't even know what I mean. These are just models that I know people look at.
I see. Yeah. Nope. I've just focused on mouse models, because the nice thing about mouse models, right, is that you can easily produce transgenic or knockout. Not easily. I shouldn't use that word so lightly. But it's much easier to use or to obtain transgenic and knockout animals, just because other groups have them, and also they're usually more translatable to humans, whereas those other species that you mentioned are a little bit less far removed. But yeah, that's a great question, and certainly people study that, but I've only studied mammalian circadian rhythms. So
okay, well, once the first cohort of citizen data becomes available, it'll be great to look through and see if there's any really interesting missense with a strong sleep.
Definitely,
a strong sleep thing that is maybe different, right? Like if it had a slightly different than the
exactly. Yep.
We had a question from Marcos, who is a parent in Germany, and he was just saying that he's noticed like a seasonality with sleep cycles of looks like summertime they see better sleep, and during the wintertime they see worse sleep. And he was just mentioning that they're in Germany where it's darker in the wintertime. And so he just wonders if there's any correlation to that, which might tie back to JR's question a little bit in terms of changes to the environment. And
yeah, exactly. Yeah, so that's again where that bright light therapy, typically in the morning, again, depending on when the child, is more or less likely, to fall asleep. So if he or she is having trouble falling asleep and doesn't go to sleep until 2 or 3 in the morning, then you could definitely use bright light therapy, which is actually a very good treatment option for seasonal, depression. And you would do that in the morning to try to phase advance your clock. So you would try to kind of align your body's clock with what you're seeing, externally. And so like you mentioned, in the wintertime, it's much more dark. So if you can try to, align your body's clock and see or observe more light, that could definitely help. But yeah, bright light therapy is huge for that, seasonal depression. Not that he has seasonal depression, but it's kind of the same idea, I think, or similar idea. That's what I would speculate, at least, as to why it's different. But again, I'm not a sleep scientist, unfortunately. Hopefully soon, but yeah.
I have it catch my attention that you mentioned tamoxifen.
Mm-hmm.
What is the effect of tamoxifen? I mean, I know
yes.
it's more as a anti-estrogen therapy.
Yeah, I'm sorry. So the tamoxifen, actually, when I referred to that, so the SynGAP het animal that we have from Dr. Roomba is a floxed mouse. And so what that means is that there are these lock stop cassettes, essentially, that you can cross to a inducible mouse. And so basically you can cross his mouse, this het mouse, to another mouse. And then when you administer tamoxifen, the only reason I mentioned tamoxifen is that tamoxifen is what allows for that restoration of SynGAP. So it's not like I didn't mean it to be like a treatment or a therapy. That's just how, in this mouse model, that's how you recover or restore SynGAP protein. Does that answer your question? I guess it was just a method of how to get this mouse model to work, essentially. And so that's the system that Dr. Gavin Roomba designed, is to, these het mice, they can function as these haploinsufficient, for SynGAP mice, but when you cross it to this other mouse line and then inject tamoxifen to allow for this Cree recombination to occur, that then can restore or rescue these protein levels, these SynGAP protein levels. So tamoxifen was just used as a way to get this mouse model, this mouse system to work. But
any other questions?
I think that, yeah, we covered the whole Q&A.
Yep, I don't see any other in the chat either. This has been great, Dr. Aten. Thank you so much for today's presentation, and really appreciated this. It was so interesting, and yeah, thank you very much.
Thank you guys.
Yeah, thank you so much for having me. Yeah, thank you. It was awesome. You guys are incredible. So I think
good luck in Boston.
Thank you. Have a great day.
Bye-bye.